So our next speaker is Dr. Haim Cohen from the Ariel University and the Ben Gurion University in Israel. And he will be speaking about the color and nature of diamonds using EPR. Please let's welcome Dr. Cohen.
What I'd like to tell you a little bit today is on trying to use a rare technique which is being not used too much in the diamond industry in order to understand a little bit better what occurred to diamonds upon treatment. And the work that I'm going to talk about today is a joint work with Dr. Sharon Rutstein, today professor, at another university in Israel, and she's the EPR specialist. I guess all of you know that fancy diamonds can have almost any color which is in the visible spectrum. And as already has been discussed, the source of color in diamonds stems from three possible reasons. One, which is the main, is nitrogen contamination in the crystal lattice, and this accounts probably for more than 90%, and it accounts for all colors. Plastic deformation, which in our estimate can come up to 3 or 5% of the diamonds, these are mainly young diamonds, can account for some brown color which is induced in the diamonds, and boron contamination, which is very rare and just now discussed, it's blue-grayish. The problems in evaluating the color in diamonds when you measure diamond, and it's mainly in rough diamonds, is that the present methods, which are UV visible spectrometer, is not very good for rough stones. Infrared spectroscopy, which is something that we use a lot in our lab, is time-consuming. Human eye is very fast, but bad compared, especially for rough diamonds, to instrumental methods. And XRD and imaging are relatively slow and expensive methods. Nitrogen in diamonds, and these are three possibilities. 2A, as discussed, no nitrogen or almost no nitrogen. Pink, which is entry and vacancy. And yellow, 1B, which is always what we call young diamonds, because all diamonds which are produced naturally, most nitrogen atoms are of the type of 1B. This is an example of a treated pink 0.32 carats, and I'm involved in some treatment procedures in a company which is called Dianer, and from it stemmed the work that we have, I'll tell you today, over here today, which is a work which started about half a year ago and looks for us very promising. And this means that though it started from kind of applied research, treating of diamonds, always when there is research, it doesn't matter if it's applied, it always comes also to basic research, which means PhD projects. If we look at this method of electron spin resonance, or sometimes called EPR, electron paramagnetic resonance, this is a non-destructive method. You measure what occurs with a diamond, nothing occurs to the diamond. When you use ESR, it's usually mainly in chemistry, and I'm a chemist. It determines radicals, namely electrons with a distinct spin, and it determines also the radical structure. And we asked ourselves, can we use this ESR spectroscopy in order to study diamonds and in order to study what occurs to treated diamond, what occurred to the structure. And if I'll talk 1.5 minutes about this spectroscopy, what ESR does, it measures transitions between electron spin energy levels. These are transitions which are induced by the appropriate frequency of radiation, and we are talking about terahertz spectroscopy or gigahertz microwave region. What we get, and you can see over here, a typical spectrum of an ESR measured. And what is important for us is that, I've listed here in red, the interactions with neighboring nuclei. You can look at radical, at the unpaired electron, and determine exactly what is the percentage of this unpaired electron on a certain atom in the crystal structure. And moreover, you can get what neighboring atoms do affect this percentage of unpaired electron on a certain atom. And you can also measure the distance between centers of two signals, which you see over here on the ESR spectrum, which consists of two signals. Operation-wise speaking, this is the two large magnets which differentiate between the two energy levels of the spins. This is the producer of the terahertz energy. What we do is we put the diamond in these quartz test tubes and put it over here and measure it, and essentially now it doesn't go. Essentially, this is the experimental setup that we have used. The treatments of the diamonds in order to induce color in them, first of all, you have to determine the right diamond which is fit for treatment. Then what you do, in some cases you use high temperature treatment. Sometimes you don't use it. Then usually you irradiate with a high energy electron beam, usually 2 MeV, 2 mega electron volt energy. And then you do another thermal treatment in order to bleach bad colors which induce or reduce the quality of the treated diamond. Essentially, you can produce any color of diamond that you want, and the diamonds that we have measured are pre-blue, a diamond which fits for treatment to produce a blue diamond, pre-yellow, pre-green, pre-cognac, and this depends on the nitrogen and type of nitrogen content and the dose which is used, the irradiation. And we produce pink, blue, yellow, green, and cognac. From each color or each pre-color, what we measured was 3 types of 0.1 carat diamonds and 3 diamonds of 0.03 carat in order to have a little bit of statistics. And let me show you the experimental that we used was infrared to measure the diamonds before and after treatment. We looked at the fluorescence, but this didn't matter too much. And the main technique that we used, as I said, was the ESR. These are the diamonds that I'll display the results, but from each diamond, as I said, we have measured 6 diamonds of 2 sizes. So you can see over here the pre-yellow and the results, the yellow, pre-blue and the result. Green, by the way, pre-blue and pre-green are the same. The pink diamond and the cognac diamond. And for example, over here, what you can see is the blue 0.11 carat diamond. You can see over here the infrared spectrum, and you see that there is a little bit, not too much of nitrogen in the diamond. This is the ESR spectrum, and I'd like you to look also at these bits. These bits means that this is the effect of the nitrogen atom on the radical, which is mainly centered on the carbon atom. What we find over here is that the unpaired electron that we detect, 60% is centered on the carbon atom, and 40% is centered on the nitrogen atom. So this means that a nitrogen atom is adjacent to a carbon atom, and we can measure the concentration of the unpaired electron on both atoms. This is the cognac, and you can see that this is a high nitrogen content. And you can see again, but over here you can see that the splitting by the nitrogen atom of the carbon radical is totally different. And you can see that in this case, 98% of the unpaired electron lies on the carbon, and only 2% lies on the nitrogen atoms. With the green, what we find is exactly the same like the cognac. What we find is that 98% lies on the carbon, 2% of the unpaired electron lies on the nitrogen. This is the pink. With the pink, the situation is much alike, but what we find is that there are two types of carbon radicals in the pink, which means that the pink, from this point of view, is a very special kind of a treated diamond. This is the yellow, and on the yellow you can see that there is much higher unpaired electron which lies on the nitrogen compared to the cognac and the green and the blue. We wanted to see a little bit on homogeneity, and over here you can see what occurs on 3 pre-blue diamonds. The funny thing is that on the pre-blue and the pre-yellow, you almost can't find an unpaired electron. The concentration is very, very low. The reason is very simple. Most of the radicals which are produced in the diamond are produced because of the electron beam irradiation. This is the production of the radical. And this is, again, if we take the green diamond and put it on several positions, we see that the signal doesn't change too much of the unpaired electron. Over here, there is a sum up of radical concentration. What I'd like you to see is that the radical concentration is very low in the pre-blue and the pre-yellow, which means, again, that the radicals are formed because of the irradiation. And over here we can see homogeneity, and we see that the pre-yellow is pretty homogeneous from radical concentration. The pink is not very homogeneous. The cognac, which is the highest irradiation dose, is pretty homogeneous. This is in a table sum up of the distribution of the radical concentration on the nitrogen compared to the carbon, and you see that most of the unpaired electron lies on the carbon atoms. Now, what is really nice is you can take the ESR spectrum, use a MATLAB, a special program, which will tell you what is the position of the nitrogen atoms around the carbon radical. And this was something that for us was very interesting. For example, you can see that with a cognac diamonds, there is a radical which is mainly on the carbon atom. However, it is surrounded via an unsymmetrical Dalton structure by 3 nitrogen atoms. The structure of the blue from point of view of nitrogens compared to carbon radicals is that there are 5 adjacent nitrogen atoms surrounding the carbon radical, but again, this is not symmetric. What we else find is something that we already knew. The difference between a green treated diamond and a blue treated diamond is only the dose. With the green, you have a much lower dose of irradiation compared to the blue, and essentially the green diamond from point of view of the structure is again 5 nitrogen atoms surrounding the carbon radical, and the only difference is the intensity of these centers. With the yellow, what we find is something which is very symmetric. We find that this is in the center surrounded by a square of 4 nitrogen atoms. The C should be in the center, and I apologize for the quality of this transparency. This is the structure of the pink treated diamond, namely a triangle where the carbon radical is surrounded by 3 nitrogen atoms. So far, we feel very satisfied with the results so far. You should remember that so far, we didn't do the right experiment. And the right experiment means you take a pre-treated diamond, you measure the ESR and the infrared, then you do the first part of high temperature and you measure what occurs. Then you irradiate and form the radicals, and again you measure. And then you do the final thermal treatment to bleach somewhat with the finest bed colors. But the one thing that we know is that as the homogeneity of the diamond is pretty good, though we can determine what is the dispersion of the nitrogen atoms surrounding the carbon radicals, and this is only with the radicals, but as the diamond is homogeneous, probably this occurs the same in all the structure of the diamond. So as I'm a professor and I don't work, the PhD students should do the work, and they are working heavily on it. And as you can see at present, the conclusions are that radicals are forming the diamond crystal mainly via the radiation process. The ESR spectroscopy can indicate the structure of the nitrogen atoms with respect to the carbon radical. ESR spectroscopy is an excellent technique to determine structural changes of the nitrogens surrounding the carbon. And the ESR spectroscopy can determine the homogeneity of N in the diamond crystal. One thing to state more, the work that I have discussed now for you is only beginning, and it's basic research. There is nothing which you can say that is applied. But as probably all of you know, there is no difference between basic and research. Basic drives to applied, applied drives to research, and from this point of view, what I can only say is that this is, for me, a very interesting piece of research that we are doing right now. The acknowledgements are to the Diana Diamonds for the diamond supply, Mr. Kahana for the color enhancement methods, and the PhD student for the data evaluation so far. And of course, without Professor Rothstein, who is doing the ESR experiments, I could do nothing. And thank you very much for your attention.
Thank you very much, Dr. Cohen. Do we have time for a couple of questions? Yes, please.
Yeah, could you elaborate a little bit on the ESR technique and how you use that in terms of structure?
I didn't, can you raise your voice?
The ESR technique and how you use that to determine the structure of the spectrum.
Well, you saw the spectra, okay? And the spectra means that the main radical is somehow splitted by the bips on the right. And in ESR spectroscopy, there is a special program which is called MATLAB, which is designated. MATLAB is not only for ESR spectroscopy, but there is a special branch of the MAT which determines what should be the theoretical spectrum. And from the determination, you can determine what number of nitrogen atoms and more or less at what distance from the carbon radical, which is the central, they are. That's why I could say that it's symmetrical or not symmetrical. And this means that from this point of view, the ESR spectroscopy is very powerful. One thing that I didn't say is that ESR spectroscopy, in order to determine radicals, is something like, I'd say, 10 to the 7 to 10 to the 8, more sensitive than infrared spectroscopy or other types. So from this point of view, it is a very powerful technique.
So just to clarify, it's a MATLAB optimization?
Yeah, it's a special, it's a special program which determine or suggest what should be the theoretical spectrum if there are 6 or 7 or 4 nitrogen atoms surrounding the carbon radical, and what are the distances, because the effect, of course, is reduced when the distance increases.
We can just take one more quick question. Yes, please.
You can speak Hebrew, Menachem.
Let's keep it in English, please.
You said a very interesting thing that the difference of the color of green and blue that you get is more time of enhancing. Did you actually do a research on how long does it take to move it from the blue to the green?
At this moment, the one thing that I can say is that from point of view of how to produce a treated green or a treated blue, you choose the same pre-diamond. It's the same diamond which fits. The only difference is that the dose of the beta irradiation, the high energy electron beam that you irradiate, and sometimes there is also different in the thermal treatment. For example, with a green color, you don't do any thermal treatment to bleach bad colors. With the blue, you do a little bit of thermal treatment. But in all, at least so far, 6 diamonds from each color that we have determined, we got the same results.
Okay, thank you very much. Let's thank our speaker.